Yes, so for a big group of patients who are fit for intensive chemotherapy, the standard curative approach still is the combination of Daunorubicin and cytarabine in the 7+3 combination, plus minus novel agents. And there are still, although this treatment schema exists or scheme exists for such a long time, or has been there for such a long time, there are still unsolved questions. And one question is whether the dose of Daunorubicin in first induction should be 60 or 90 milligrams. And the second question is, for patients having a good response after the first induction cycle, do they actually need a double induction, or can we get rid of the second induction cycle if they have good response? We try to answer those two questions by two randomizations in one single study that was called DaunoDouble. And the main results are that 60 versus 90 milligram does not make any difference, neither regarding early responses, but also not regarding CR rates, neither survival. So it seems that there is a tendency for slightly more toxicity in the 90 milligram arm, but in terms of efficacy, it's almost exactly the same. So our conclusion from that trial is that, from that randomization part, is there is no rationale based on our data, and there have been data in the past from the British NCRI 17 study. There is no rationale to use 90 milligram of Daunorubicin anymore. And the second part of the trial looked at patients with a good response after the first induction cycle, and the randomization was between having a second induction cycle or no second induction cycle. And we could show that the CR rates are almost identical in these good early responders, and there is a slight difference or slight advantage for double induction in terms of relapse-free survival in these good early responders. However, that is borderline significant, and if we look at multivariable analysis taking into account other prognostic variables, the difference disappears. And when we look at overall survival, finally, there is no difference at all whether these good responders received 1 or 2 induction cycles. Therefore, the conclusion for this part of the trial is, in good responders after first induction, there is no need for a second induction cycle.